function. But all what we can do for those is to try to not allow the damage to accumulate in them too much until they become senescent. So one way to do that is to reduce a damaging agent that may affect them, or to try to time remove damage from them as it accumulates, to prevent it from reaching a threshold that induces senescence. Now, even better is to try to partially reprogram these cells, because now the technology exists. So this can be even a more compelling approach to partially reprogram them to an earlier stage. The reason is that these cells now, the young cells, they can repair the damage by themselves. So it makes it even more compelling. So of course, the best way is to try to combine one or two of these approaches to really minimize the damage to a maximum. And that's what we are doing in our reconnaissance program. We are targeting senescent cells by elimination with senolytics. We are also trying to reduce damage and for the cells who will not even become senescent. And we are trying to partially reprogram functional cells, so they will be able to repair the damage by themselves. For the sake of time, I will focus only on the damage reduction part. And so for this, as you know, senolytic field have taken last few years, and there's a lot of senolytics, mostly for the discovered from cancer. The most common is navitoclax. But most of these senolytics, they target only one pathway and or one molecule in that pathway, and so they're not expressed in every tissue. And in the case of navitoclax, for instance, the target BCL-XL is expressed in other cells and even in the platelets, and that can cause thrombocytopenia and side effects. So this represents a major limitation for using this kind of drugs in the clinic. There's another way to go around it, is to make a prodrugs of navitoclax, and a group in Spain, they add a molecule galactose to it. That galactose actually is substrate for the enzyme that characterizes senescent cells, which is senescence-associated beta-galactosidase. And so when it is in cells that do not express this senescence-associated beta-galactosidase, that prodrug will not be active. So even in the platelet themselves, it will not be active. It's active only in cells that express this senescence-associated beta-gal. But we tried in vitro, and it works fine. It does actually have a preference for senescent cells, almost like navitoclax. The only thing is that when you try this in vivo, it's toxic also. And the reason is that beta-gal, this enzyme, is expressed in virtually every tissue, and it can be activated by even a small variation of pH. So yeah, it may not be translatable to the clinic also. So we are now working on a different type of prodrugs, which are activated by completely different mechanism of action, and we are making progress on that, and I hope I can give you more update in the next time. But just take you back to this problem that actually the Asian research field have, is that there is the lack of markers and the lack of targets to target senescent cells, or even to identify them. And this is it has been a problem for a long time, and there's a lot of studies that has been conducted, to compare the profile of senescent, non-senescent cells. A lot of omics has very big data studies, But with all this, we don't have a marker yet. And the only targets that actually are targeted by senolytics, they are expressed in only a few type of cells and few tissues. So they're still unlikely really to affect, to benefit the whole body. So now, if the goal is to listen or to lower the damage accumulation in the body, for instance, let's say by removing senescent cells, which are actually damaging agent by themselves because they're secret cells, then removing a damaging agent molecule that is endogenous, basically endogenous toxin that is produced within us, that if we can remove that one, then maybe this can lead to the same end, to reducing damage in the body. And one key molecule that it's damaging also, that has been largely overlooked for its role in aging, is heme. As you know, heme is a molecule that is very important for life, for breathing. It is associated with hemoglobin, which is found in red blood cells. And hemoglobin and heme, of course, play a key role in oxygen transport from the lung to the rest of the body, and it plays a role in many areas of metabolism, mitochondrial function, energy production, and many enzymes that utilize heme. However, heme is very toxic, and if it is not well controlled, it can precipitate the end of life. And there are examples for that, which is hemolytic disorders and hemorrhagic crisis. We have found that for the first time, I believe that heme is increased in senescent cells, and we use two different assays, and they all show the similar results. Here, this is a kit from EBICAM, and this is a probe that we have identified that is highly specific for heme, for labile heme, and we found that it is increased in senescent cells, and whether endogenous one or if we add heme, it's also they retain it, which is very interesting. So for us, this initiate a whole series of investigation, and we're asking a lot of questions that we're investigating right now. First, of course, where does heme come from, and is it toxic? What are the short term of its toxicity, long term? Is it the cause or the consequence of senescence? How to prevent the damage that is caused by heme, and the most, of course, the goal is can we target heme to treat aging or the associated diseases. And so for this, to give you a little bit where this come from, I want to introduce you to another type of senescent cells, which are senescent red blood cells. They are actually the major source of heme in the body. Each RBC contain about 280 million molecules of hemoglobin, and if you multiply that by 4, which is because every hemoglobin contain 4 hemes, so almost 1 billion of heme molecule per red blood cell. Red blood cells, they live about 120 days, and then they become senescent, and then they go the process of degradation that I will discuss later. On the daily basis, there's almost 20 billion senescent cells that are cleared every day, and that's a lot. That's really a lot of senescent cells. So now, if we compare that with other senescent cells, it looks like the senescent RBCs are the largest fraction of senescent cells in our body, and also they release heme, which is much more toxic than SASP. And another particularity which is interesting is that they are more dangerous when they are dead than when they are alive. So that means senolytics should not be, of course, used for RBCs. And by this way, actually, the senolytic term needs to be a little bit modified or revised, because it should specify senescent cells that are non-RBC. Other, if it is hemolytic for RBC, then it's not good. So this kind of comparison between two senescent cell type bring a lot of question that actually needs to be addressed. So in a normal situation, when red blood cells become old, it gets taken care of by macrophage, and it's recognized, and it gets on the phagocytized, and the heme with it is degraded, and everything is controlled, and the iron go to ferritin, and everything is under control. But there are cases the hemolysis can occur only also by itself, when macrophages are overwhelmed, and this leads to hemoglobin, and whether tetramer or dimer, but and then of course ultimately to heme. But fortunately there are some protein in the plasma such as haptoglobin that take care of hemoglobin dimer, and hemopyxin, which take care of labile heme, and then bring them to either macrophage or to Kupffer cell in the liver to be degraded. But these protective mechanisms become overwhelmed during hemolytic diseases or a hemorrhagic crisis, and this, of course, lead to serious consequences. Now, there are examples of hemolytic conditions. For example, sickle cell disease, where basically hemoglobin is mutated, and red blood cells change morphology, and that can accelerate actually their hemolysis. And basically these patients, they don't live more than around 52 years. So their lifespan is shortened because of this excess of labile heme in their system. As you can see, the normal amount of labile heme is about 10 to 20 micromolar in the plasma, but then it becomes 50 in intermediate, and then high, more 100 or more micromolar in the disease patients. There are also hemolysis that can happen, like intraventricular hemolysis in preterm born infant. And same thing, the level of heme in their CSF, it's way too high in first few days, but then it regresses, and the survivors, they have very strong sequelae that stay with them their entire life. So whether it's chronic exposure to heme or temporary, there are consequences that can stay for a long time. There are, of course, many causes of hemolysis, not only sickle cell, but malaria and hemorrhagic stroke, sepsis, a lot of other things, even viruses and infection, and certain medications that they also induce hemolysis, including the pain medication that we take very often, some antibiotics, and environmental factors, airborne particles, smoke, pesticides and insecticides, and all kinds of things. So you can imagine, even a normal person, like a healthy person, we are exposed to at least to the one that are highlighted in blue here, kind of frequently during our lifespan. So every time there is extra hemolysis, that damage continue to accumulate in the body over time, and it can be harmful at the end. With regard to hemolysis with age, there is not much has been done really so far, but this paper, for instance, they talk about the exhaustion of macrophage with age. So they cannot, their ability to phagocytize the red blood cells diminish. Therefore, red blood cells, they just burst in the blood, in the vessel, and release labile heme in the circulation. And on the right, you can see that in this study, they show that aged mice have more labile heme in their blood than the young one. And as you can see the value, which is about 50 micromolar in the ages. That's the same, almost the same one that we show here in some patient with hemolytic diseases, suggesting that as we age, probably we will probably start developing this phenotype as this hemolytic patient. Another thing is that hemolysis do not happen only with age, but it can happen throughout our lifespan, even when we are young. Because this paper here, this show that actually hemolysis can happen just by sheer stress. Just the cells bind to the matrix of the spleen, and just by sheer stress, they can release their heme into the bloodstream, regardless of whether macrophage are exhausted or not. So yeah, all this lead to the accumulation of heme and the associated damage throughout life in the body. And heme, it is there's a lot of literature that shows that it's very toxic and harmful itself. So it does it in many ways. The first one is basically it can contribute to the lysis of RBCs themselves. When there's more labile heme outside, heme is lipophilic, so we just go and bind to the plasma membrane and lyse this more red blood cells. So it creates more like a cycle. If in the vessel, it goes to binds to because it's lipophilic, it binds to LDL and oxidize them. And as you know, LDL cause atherosclerosis and other things. It can bind. It can induce platelet aggregation. It can inflammation. It can just bind to any cell in the body, and because lipophilic, it goes to the plasma membrane and there's a lot of RAS and lipid peroxidation, and that cause a variety of damages into the cell. We have shown that in vitro heme can induce proliferation arrest, can even induce senescence, and by comparison with iron itself, at the same amount, heme is more toxic than iron. And we have shown that not only with the cells, but also with activation of probes, with other things, always we found that heme is more potent than iron itself. This is another cell type, which is UVEC endothelial cells, which are actually the one that in vivo are the most exposed to heme. They are very sensitive to it. They can stop proliferating. It can induce inflammation molecule in them very quickly, like AR1 beta here. And if you incubate it a longer time, it induces senescence in them. So it is toxic and can contribute probably to age-associated disease based on this finding. So of course, there's a lot of literature that demonstrate toxicity of heme, but the question is what to do about it. Can we target it? If we target it, can we reduce these diseases, whether short-term or long-term effects of heme? And that actually require, if you want to target it, we need to have a good sensitive method to detect it. And we have developed one. We have found this probe that is highly specific for heme. As you can see in the blue bars, the peak is about 0.1 micromolar. When in comparison with iron, the peak is not even obtained even after 5 micromolar, which is like almost 500 times the peak for heme. So we use this specific assay to screen for FDA-approved drugs to see if we can find some molecules that can scavenge heme, that target heme. Before the screen, we optimize the assay, and also we wanted to know, because we will be using heme for many weeks and during the screen, we want to know whether the stability of heme is good. So we used one sample that was thawed a few times, and we were using it for a long time. Another one fresh, and with old heme with the probe or new heme with the probe, they all work fine. And the old heme alone, no signal. The new heme alone, of course, no signal. The buffer, no signal. The probe, no signal. Only when we incubate the heme with the probe that we see the signal. And the signal to baseline ratio was very good, so we moved and we start screening. Right now we are halfway. We have another more than 1,000 to go, compound to go, but we already having a good selection of hits, and we're in the process of validating them in an orthogonal way, completely different way. And here I just show you some top candidates, but we have a lot of options actually. This is, you can see the control left, the cells not treated with anything, and the second row, the red one, are treated with heme. So there's a lot of damage, as you can see, cells damage, and a lot of them are dying. But when we treat them with any of these 4 drugs, they don't die. So it's really protected. And that's very, very encouraging. It was a good finding that we have made. And I think this drug will have applications for the treatment of hemorrhagic diseases, hemolytic disorders, and also chronic diseases that come from this hemolytic disorders, or that accompany aging itself. The good thing is that these drugs are all FDA-approved drugs. That means their safety profile is well established, and so we don't need to go through all that to prove it. So if we can prove this protection in mice, in an animal model, that means it can be translated fairly quickly to humans. So so that's the next step that we will be trying. One of them is to try these compounds on sickle cell disease model. These mice, they live about 9 months. 9 months, sorry. So that's a good age that we can do experiment, and hopefully we can extend their lifespan. And then we will also try it in a natural aging model. We will use mice that are about 18 months. And if it's validated in this mice, then as I mentioned, these drugs are FDA approved, so they can be translatable to human fairly quickly. So that will be the next step, the ultimate step, of course. Yeah. So with this, I want to conclude that labile heme, whether it's a result from physiological stress or stress-induced or pathological hemolysis, it is a potent damaging agent that contributes to the onset of progression of chronic diseases. So the development of approach to reduce labile heme or its damaging effects may have application in not only for treating crises like hemorrhagic crisis or hemolytic diseases, but also aging diseases. And if they approve drugs, that's the one we're screening, that protect against heme damage, may have application and can be easily repurposed to treat aging-associated diseases. And with this, I would like to thank the SRF donors and project sponsors, the SRF board and the senior team for all the help, and the research colleagues and all the entire staff of SRF, of course. And last but not least, my wonderful team of members that are really working very hard to make this possible. And thank you very much. And if you have any question, I'm ready. Thank you.
Thank you, Hadi. That's wonderful. Thank you for the amazing work you're doing for us and for everyone out there. Yes, we don't have time right now for questions, but you can find Dr. Riba around the room and ask him any questions. He will be all yours. And I'm going to introduce our next speaker now. She is our dear Dr. Amutha Bhuminathan, and she leads our MitoSense program, which is one of our oldest programs at our research center in Mountain View. Her presentation today is titled Current Approaches in Mitigating the Role of Mitochondrial DNA in Aging and Age-Related Diseases. You can read her presentation summary and bio on the events website. And welcome to the stage, Amutha. Good luck.
Hello, everyone. Can you hear me? Oh. Hello, everyone. Can you hear me? Sorry. Sorry. Okay. Yeah. Hello, everybody. Thank you for that great introduction, Maria. MitoSense is a long-running program at Sense Research Foundation, and today I'm going to give you some highlights on what our progress has been on this program. We heard some great talks yesterday and today about some of the methods that we are addressing. How can we damage control for drivers of aging? We heard about how we can remove senescent cells. Yesterday, we heard about how we can have tissue replacement. One of the drivers of aging is mitochondria and its role in maintaining cellular functions. We have been working on a project called allotopic expression of the mitochondrial DNA genes. I'll tell you why this is important. In addition to the nuclear genome, mitochondria are the only organelles that come with their own DNA. And this is just a glimpse of the multitude of mutations that can accumulate within a human body. And this is from mitomap.org and the several diseases that come with it. It's estimated that there's about 500,000 people diagnosed with primary mitochondrial DNA diseases worldwide. However, 1 in 200 is born with a mitochondrial DNA defect. This week is actually World Mitochondrial Disease Week to highlight the importance of this important aspect. And in fact, several national monuments will be lit up green for advocacy and funding and how we can find cures for this disease. So in my lab, we have two main projects ongoing. Of course, the allotopic expression of mitochondrial DNA is a long-running project, and recently we've started a small molecule approach to modulate the organelle as a whole. And I will talk about this project and what is our progress in this aspect as well. However, in the larger research field, people are exploring different avenues and different aspects, like how can we modulate mitochondrial biogenesis. External organelle replacement has come. Recently there is a lot of activity in this area. There are several startup companies that have started with this approach. And of course, there are small molecules that mimic exercise. Antioxidants are a great line of treatment for mitochondrial diseases or mitochondrial dysfunction, but they are just antioxidants. They only manage symptoms. They are not able to reverse any of the damage that's caused to the mitochondria, and several supplements. So what is allotopic expression? To understand this, maybe I'll go to the next slide. This is the life cycle of mitochondria, and if you imagine, like, this prototype structures that you observe on electron microscopy, like tubular structures, once they attain a certain mass, then fission or division of this mitochondria occurs within the cell, and this divided mitochondria actually gather proteins and all the other biochemical components to again grow. However, this growth is dependent on two genomes. The nuclear DNA brings in more than 1,400 genes, that the protein, the products of 1,400 genes that assemble into the mitochondria. However, the mitochondrial genome in humans, it's about 16.5 kb, and it brings 13 important proteins, all parts of the OXPHOS relay, and that's how they are made. And then this cycle continues. On the right, I show, as an example, electron EMs of a typical mitochondrion, and the far right is the one from a cardiac myocyte. You can see how invaginated the inner mitochondrial membrane is, where the OXPHOS resides, and the level of the number of these small molecular machines that you need for the actual function of this organelle. Now, going back, what is allotopic expression? Allotopic expression is placing a wild-type version of the mitochondrial gene in the nuclear cytosolic compartment. So we're placing a gene in a location away from its natural location, and then it's efficient. And the protein products are formed in the cytosol and redirecting of these protein products to the mitochondrion, and specifically to these OXPHOS complexes. Okay, it seems at the outset, it seems very easy. Okay, the organelle is able to do 1,400 genes. It's already targeting to different parts of the organelle. How difficult is it to do 13 more? However, in reality, it's much more challenging than we think. But of course, the other question is, why allotopic expression, right? Why can't we go and edit all the mtDNA within the organelles itself? So there are some fundamental differences between the nuclear genome and mitochondrial genome. Of course, the nuclear genome, with its advanced structure and properties, it's protected by a protective covering, and the genes are actually packaged into chromosomes and proteins that protect them when they are not dividing, and the repair mechanisms are much more advanced than the organelle DNA repair mechanisms. It is away from all the byproducts of this energy generating machine, ROS. It's away from the nuclear genome is away from that. And recent advances, it is amenable to editing. Like, it's only 2 copies for any given gene. If there is a mutation, it is possible to edit using advanced techniques like CRISPR and so on. However, the mitochondrial genome is, of course, it's a naked double-stranded circular structure. Although not all the information is known, but it's believed that there are no protective proteins. Also, because of their replication, so the mitochondrial DNA replication and the nuclear replication do not coincide. They are independent. However, because of the need of so many organelles and their function, because of the rate of replication and the errors introduced thereof, it is estimated that mitochondrial DNA accumulates 10 times more mutations than the nuclear genome. And it is, of course, very close to all the byproducts of the energy generating machinery. And if you want to edit, CRISPR is not very efficient for mitochondrial DNA editing, or recently there have been some advancements in this angle. However, for any meaningful editing to happen, you have to achieve more than thousands of edits per cell, and that can give you the enormity of what we are asking here. So our idea is to place a backup copy of these 13 genes in the nucleus and mimic the 1400 other genes and see if we can retarget these protein products into the organelle. So what are the steps required for successful allotopic expression? We published this review article in 2022 where we highlighted all the challenges and advancements in this field. At the bare minimum, we would have to realign the mitochondrial and nuclear language. So the mitochondrial DNA is of prokaryotic origins, and its codon usage is significantly different. It's different than the universal codon usage used by the nucleus. So at the bare minimum, we have to realign the codon usage, otherwise you wouldn't get productive products formed. All mitochondrial proteins also have a targeting sequence. For many of them, it is at the N-terminus, but for some of them, it could be internal. But from an engineering point of view, it's easy for us to put it at the N-terminus. So you need a mitochondrial targeting sequence. And then, of course, the general concepts of how you can efficiently introduce the gene construct into the nucleus, and then you would test for functional rescue, and if we can mimic it in vivo, and what is the safety of this procedure. I'm going to take you through some of the advancements that we have made in this aspect. Of course, just to highlight, there are 13 genes in the mitochondrial DNA. There are 7 in complex 1, 1 in complex 3, 3 in complex 4, and 2 in complex 5. And as a prototype, this is the construct design. You'd have an appropriate promoter. You would have a mitochondrial targeting sequence. We have adjusted the sequence. I'll come to what is codon optimized in a little bit. You either have minimum correction in the code, or the codon optimize the sequence. If needed, if we don't have very good antibodies, you would need a tracking signal for it and appropriate terminators. With this concept in mind, we use patient-derived cybrids as model systems, in that these are homoplasmic for a mutation in any of the genes, and because this is a very good model system, so where we can really track if the allotopic constructs that we are expressing are useful, and if they are integrating into the complex and bringing back function or not. So I will just highlight. We published these 2 papers recently where we showed in vitro models that for the genes ATP8 and ATP6, we were able to rescue function in a null cell line completely devoid of the ATP8 protein, and we were able to recover several functions. Now, subsequently, we tried to scale this up for all 13 genes. We utilized something called codon optimization. In addition to mere codon correction, we also utilize prediction tools that will design the codon usage for all the amino acids. Of course, this is a commercial algorithm, but what we were able to show in this paper is that just by codon optimization, you can practically produce protein products for all the 13 mitochondrial DNA genes. However, at the functional level, not all of them are working in our hands, and I will talk about some of the challenges that we are facing there. Now, progressing with the genes that did work in our hands, ATP8 and ATP6 partially, we wanted to test the efficacy of this allotopic construct in vivo models. Of course, I have to highlight here that there is a paucity of research models in the field. So, you know, there is, luckily, there is one model available for the ATP8 gene. This is not a null model. Of course, the mice will not survive. They will not thrive if it's a null model. There is a mutation at the 5th position of the protein, and they do have they have mild ATP hydrolysis defects. However, one other thing that we discovered during this study is that some of the reported phenotypes were not replicated in our hands. The reported phenotypes were anxiety and other features, but of course this is a behavior feature and which is hard to replicate from lab to lab. Nevertheless, we used this as a biochemical model to compete. Is the allotopic ATPase that we are sending into the mitochondria, how efficiently can it compete with a mutant protein, which is probably the disease status in populations. So one thing to remember is mitochondria are maternally inherited. And please remember this, because when I explained how we generated this model, it's important. What we did is we injected fertilized eggs with the allotopic ATP8 transgene. This was done in a maximally modifiable mouse. This is developed by Applied Stem Cells and was funded by Sense Research Foundation. We used this model to introduce a single copy of the ATP8 gene in a safe harbor locus. We put this in the H-ROSA 26 locus of the FVB mouse. So the FVB mouse is, I have to tell that the FVB mouse, the original FVB mouse, has a lot of mutations in its nuclear DNA and the mitochondrial DNA. But Dr. Ibrahim's group from Germany, what they did is that they backcrossed this FVB mouse with the C57 mouse to eliminate most of the nuclear DNA mutations. They backcrossed 10 times. So the resulting model is mouse with only the mitochondrial DNA mutations preserved. So once we introduce the embryos with OATP8 transgene into the maximally modifiable mouse, we will generate mito mouse with a nuclear ATP8 gene in the H-ROSA 26 locus, and we take males from this and we cross with the females from the ATP8 mutant mice. So all the progeny, because of mitochondrial maternal inheritance, all the progeny is going to get the mutated mitochondrial DNA. So we cover the transgene, a copy of the transgene is in the nucleus, whereas all the mutant mitochondria will be present in the progeny. We confirmed that, yes, we have placed a single copy. You can see that lane 1 is transgenic and lane 2 is non-transgenic. There's a single copy of the gene, and we have also validated this with next-gen sequencing. And then what we did is we characterized, yes, how well does this transgene fare in our hands. What we observed is that in both, we placed this transgene in wild type and in the empty FVB models. So the ATPase transgene is expressed across several tissues, brain, liver, heart, skeletal muscle, everywhere. And for sake of time, I'm not going to go through all the functional characterization. What we were able to show is that, yes, this transgene protein product efficiently integrates into the OXPHOS complex. It does not impair function adversely. When I say this is because we were not able to replicate some of the behavioral phenotypes that was reported previously. However, this transgene at least doesn't impair any function. So we tested this using ATPase assay, oxygen consumption, metabolic cage assessment, and all the other tests. We also confirmed what is the immune profile. So we did a cytokine array. It is tolerated with respect to immune profile, and there are no drastic changes in the global protein profile. We did this using mass spectroscopy. And this transgene was also expressed over 1 year in the mouse, and it is faithfully transmitted up to 4 generations with no change in fertility. So all this shows that placing a mitochondrial gene in the nucleus, at least for ATP8, is tolerated very well, and it can be inherited as well. Okay, that is for the ATP8 gene. We've also implemented this codon optimization and expression for one another gene. This is the ND4 gene. We were able to show that there is rescue in function for a Leber's hereditary optic neuropathy model, this gene is also working to a good extent in our hands. However, some other genes are not working as well. So our current strategies are to introduce rational libraries. Like, one major factor that all these 13 proteins may be hard to import into the organelle is their hydrophobicity. So we are implementing some computational approaches to reduce the hydrophobicity, we've implemented this with respect to the COX-2 gene. I'm showing here that, COX-2 allotopic protein is practically not processed. But when we reduce the hydrophobicity, we know at the biochemical level, yes, it's processed to what we want to see. However, COX-2 has 2 transmembrane domains. What we also showed is that unless you reduce TM1 hydrophobicity, it's not useful reducing TM2 hydrophobicity. However, at least yet, we don't see any functional rescue. So we are implementing several other computational approaches to bring back function for these allotopic genes. Our current strategies are around, finding the optimal mitochondrial targeting sequence and also the appropriate coding sequences, how we can change to bring function. With that, I'm going to switch to the second project that I was talking about. This is mitophagy, and again, coming back to the life cycle of mitochondria, when it's a cycle of fission and fusion. However, when fission happens, parts of the damaged mitochondria are selectively targeted for degradation by the recycle mechanism within the cells. That's they fuse with lysosomes, and then that's a very specific organelle-specific degradation and recycling of mitochondria. Of course, mitophagy can be induced several different ways. You can target the membrane potential, PINK, PARK in several modes, lipid modification. However, And we want to utilize something that's like the energy status of the mitochondrion, because then it can be mutation agnostic. Just to highlight how mitophagy and age-related diseases are connected, in sarcopenia, it's observed that this recycling mechanism is aberrant, and therefore you get degeneration of the muscles. Um, so we are using two models to test our small molecule approach. One is the common deletion cell line, where you have 6 mtDNA genes that have been removed. And another one is a point mutation, a Leigh syndrome line with a specific mutation in the ATP6 gene. What we expect here is if we can increase the wild type mtDNA, if we can decrease the mutant mtDNA load, and if this in any way restores OXPHOS function. When we treated the common deletion line with our compound, it's called SRF1 here, for 28 days. This 28 days is because mitophagy occurs anywhere from 3 days to 15 days. So we wanted to give at least 2 cycles of mitophagy to undergo before we analyze the cells. So what we can see is that compared to DMSO treated cells, there is after 28 days, there's about 25% decrease in mutation load. This is using qPCR. And for a functional assay, we did the impedance assay. That's viability on galactose, which is restrictive for oxidative phosphorylation function. And if you see DMSO galactose is here, whereas the SRF plus ROC2 treated samples regain significant viability. And this is again similar for the Leigh syndrome line. Again, we see a 25% mutation load decrease after 28 days. And this one showed much significant growth on galactose. Kate is now working on, the gold standard in mitochondrial function oxygen consumption assay she's done for the BH2 cells. What you can appreciate here is that the oligomycin sensitivity and the rotenone antimycin sensitivity is significantly improved in the SRF plus ROC2 treated cells, which is similar to, in fact, 143B cells. And there is a similar increase in function, what we see to the mtDNA load. We are still doing these studies need to be quantitated more, and we are repeating this most, but I just wanted to share this exciting data with you.
Ammuta, I'm here with you on stage just to tell you have a minute to wrap up.
Okay. So yesterday you heard a possible mode of action here. All these compounds were identified as ATP, the ATP synthase inhibitors, and there is a suggestion that it could act through ATP-IF1. However, our genetic studies suggest that this may not be the case. It might be an upstream regulator. So we are still working on the possible mode of action here, but these are promising results. With that, I just want to thank my team, a great team. Without this, none of this would be possible. Kate is working on the mitophagy project. Bhavna is working on the gene therapy aspect. Jonathan recently joined the team, and he's also working on the gene therapy aspect. Marek did many of the computational aspects. We hosted 2 summer students, Anaga and Sahiba, over the summer, and our postbac scholar is Simon. And this is our funding and our collaborators. Thank you.
That was wonderful, Ammuta. Thank you so much. Big virtual hug. Thanks for all you do. And yes, everyone is sending hearts and likes. So we're going to take a break, 30-minute break, and we will see you again at 11. Please have fun and check out our booth, student booth, and we'll see you back with our student presentation at 11. Thanks.
Hi, everyone. I'm Lily Fishman. I'm the Director of Academic Affairs here at SENS Research Foundation, and I wanted to thank you all for being here and also thank our students for their work both this summer and this past school year and for their time now presenting that work. I want to encourage everyone to take a stop by the various posters that are around the conference hall, take a look at the brilliant work that our students are doing, and our students are rotating near their posters. So please, if you have any questions, feel free to click on the students and ask them all the questions. I'm sure they're very excited to talk about their work and their time this past summer or school year. Thank you all again, and I will see you around. Hi, everyone. I'm pleased to introduce our second student presenter, Danielle Van Sover. Danielle's a senior at New York University, where she's majoring in biology. Danielle's passionate about aging research because she seeks to define the absolute truths of biology. I know she's learned a lot this summer about how the passage of time might be absolute, but diseases and processes of aging are not. Danielle's previous experience focused on regenerative medicine and mesenchymal stem cells contributions to skeletal muscle and adipose tissue. Danielle's interest in regenerative biology was a perfect fit for her summer placement here at SRF under the advisement of Dr. Hadi Riba. Danielle will be speaking today on heme, the toxin in our blood, investigating heme toxicity in an aging context. Thank you for your time, Danielle, and I can't wait to hear more about your work this summer.
All right. Hello, everyone. My name is Danielle Van Sover. As Lily said, I'm currently an undergraduate at New York University, and I'm studying biology. And today I'll be sharing my research from this past summer as a scholar at SENS, investigating heme toxicity in an aging context. So to provide a bit of background, I want to briefly start off with what it means to age. So the phenomenon of aging, looking at it through a biological context, is often generally defined by the buildup of damage. Over time, our bodies undergo what could be described as wear and tear. The many physiological systems and intricate mechanisms key to our survival gradually decline in function. And in the face of many pathologies and diseases, this decline only accelerates. And the sources and symptoms associated with this decline include many familiar phenomena, such as genomic instability, telomere degradation, epigenetic changes, and many more. So there are many factors that contribute to the aging process. One such factor investigated heavily at SENS is that of senescence. It's widely understood that our cells undergo a variety of changes, and this is generally dependent on the locale of a cell and tissue state. And senescence is a particular cell fate of great interest, as it's found to be highly associated with aging. As we age, senescent cells increase. A senescent cell is one that no longer divides, yet it still remains alive, often described as a zombie cell. Senescence does have a necessary function. It protects against uncontrolled cell growth, as seen in cancer. Yet in spite of this function, senescent cells also resist the body's usual system of disposal and often linger in their environments where they secrete high levels of SASP factors. These are harmful compounds that cause further damage to surrounding tissue. And this summer, I had the opportunity to work under the mentorship of Dr. Hadi Riba with the Replenisense Lab. And a big question that Repleni is investigating is not necessarily how we can remove senescent cells, but whether we can actually delay the onset of senescence in the first place, and therefore maintain a cell's functionality and capacity for longer. Now, this would have to apply to the many processes that occur in the body, all of which possess many distinctions from one another and would first need to be comprehensively and precisely defined. So currently, the lab is investigating one specific phenomenon that is highly relevant and occurs in all of us on a regular basis, and that is hemolysis. And the lab is studying this and the many implications associated with it as significant sources of damage and aging. So hemolysis is the destruction of red blood cells. It's a completely normal, steady state process that occurs in all of us, since the body needs to constantly replenish old and damaged red blood cells. Now, a single red blood cell contains approximately 260 million hemoglobin molecules. These are the pretty well-known proteins responsible for oxygen transport through the bloodstream. During hemolysis, however, these hemoglobins are released from the cell, at which point they can be oxidized or broken apart, and in turn, they release a compound known as heme. And so heme, it's an essential compound that performs many functions associated with gas exchange, energy production, and signal transduction, among other functions. Yet when completely exposed from the red blood cell and no longer housed within a protein, heme has been found to be highly toxic due to its hydrophobic properties and high reactivity. It promotes oxidation, changes the structures and functions of essential proteins, and generally causes significant cell and tissue damage. And in the case of many blood conditions, including sickle cell anemia, thalassemia, and even during blood transfusions, hemolysis is upregulated, and red blood cells are actually destroyed much faster than they can be replaced. And in these cases, the free heme pool that's established continues to accumulate, and its effects become increasingly severe. So the central question that my project is hoping to answer is, can we determine whether heme is a source of cell damage and senescence? And if so, can we then develop a targeted therapy to reduce free heme in the body and ultimately minimize its effects? And so in order to begin addressing these ends, my project focused on two central aims. The first is to test heme toxicity on cells of interest, observing both the short-term and long-term effects. And the second aim of this project was to test certain compounds as potential sources of heme neutralization. I focused primarily on albumin, the most abundant protein found in the plasma, and therefore one that would very likely interact with free heme in a hemolytic event. And albumin is known for its antioxidant properties, so a goal was to test those in cells against heme. So to test the short-term effect of heme and the protective properties of albumin, IMR90s and HUVECs, are cells of interest, were cultured and then treated with heme and albumin in a dose-dependent manner. At the endpoint of a treatment, cell viability was measured using a luminescent ATP detecting assay. So essentially, a greater signal indicated a greater amount of metabolically active cells. So these are the results of this experiment in IMR90s. In the two sets of images on the left, we can see the two extremes of heme treatment with no added albumin. So the top image depicts healthy cell proliferation and extremely minimal cell death. And in the image below it, containing 10 micromolars of heme, there's an evident reduction in cell survival in comparison. And the four images on the right all contain 10 micromolars of heme, yet they also contain increasing amounts of albumin. And as we can see, with no albumin included, cell survival is significantly reduced, yet already at 25 micrograms per ml of albumin, proliferation picks up, and by 100, cell survival is nearing the amount seen in the original control. And these two graphs depict these results. The graph on the left displays this treatment in IMR90s, and on the right in HUVECs. An initial view of these gathers that as heme does increase, there's a reduction in cell survival, yet adding albumin treatments in an increasing manner to all heme doses appears to allow cells to recover. So this is a positive sign for albumin and begins to confirm that it has some protective properties against direct heme exposure. The second aim was to determine the long-term indirect effect of heme. Part of heme's toxic repertoire is used copper sulfate and the other hydrogen peroxide and heme. After incubating the protein with either compound, oxidation was verified using a detection kit, and these samples then underwent immunodetection to be visually confirmed. This was perhaps the most challenging aspect of this project, since no oxidation protocol had been previously set. So there was a lot of trial and error that went into this endeavor, but it was a welcome challenge, and I definitely learned a lot from it. So in terms of results, these depict one such attempt at using hydrogen peroxide and heme to oxidize albumin and subsequently treat IMR90s with the oxidized protein. So the Western blot image contains samples of unoxidized and oxidized albumin with and without the use of the detection kit. And while some differences are observed in the oxidized sample, I'd conclude that these results are far from final and that a more optimized protocol be set given more time. In regards to cell treatments, this graph exhibits the effect of oxidized albumin on IMR90s as seen by the red bars. And while there appears to be a reduction in cell viability relative to the albumin control, there are additional factors to be considered, such as heme, which could have been acting alone. So from here, I can only conclude that while this experiment is moving in the right direction, it requires additional adjustments for more optimized results. And so given additional time to dedicate towards this project, next steps would involve continuing to optimize a protein oxidation protocol, as mentioned, and continuing to assess both directly and indirectly heme damaged cells in the context of senescence. So including further testing for characteristics of aged and or damaged cells. And this would include SA beta gal activity, certain SASP factors, and increasing reactive oxygen species, for example. And by continuing to define heme-induced damage and doing this research and modifying potential solutions such as albumin, we'd be able to more accurately target the buildup and accumulation of this harmful molecule in the body and reduce its consequences not only in those suffering from various blood conditions, but in all of us experiencing the disease of aging. And to conclude, I just want to briefly express how the concept of ending aging or even delaying it has always seemed to me like a substantial and even inconceivable endeavor. And this is because we've been accustomed to believe that aging is a forever fixed fact of life, but who can blame us? And yet my time at Sense has shown me the value in altering that perspective and viewing the aging process as a disease that can be tackled like all others. And so I want to end by saying that the endeavor to minimize the risk of age-related disease and to improve our health spans and our lifespans in the long run is one very much worth the investment of time, effort, and optimism. And I'm very grateful to everyone at Sense for the opportunity to contribute to this effort and become even more excited about the future. And I'd just like to quickly thank and acknowledge the entire ReplenisSense lab for the mentorship and guidance in navigating this project, and the education department for this very rewarding experience, and everyone at Sense. So thank you.
Thank you so much, Danielle. I'm getting a flood of comments both through this platform and through, my email saying how much they appreciate your perspective and your reminder that aging is pathological and can be tackled. So I'm glad to hear that that paradigm is changing. It's fantastic to hear. We do have a couple questions in the chat. One of them being, how do the levels of free heme and albumin compare to the levels to which cells in solid organs would be exposed outside of hemolytic crisis conditions, such as having a stroke? Do you mean to repeat that?
Sure.
Okay. They're asking about the levels of free heme and albumin in solid organs in a normal environment compared to the levels of heme that you're treating cells with.
Right, I see. So I believe the concentrations that we were testing were matching the physiological conditions in a normal scenario. And I guess you could say a hemolytic event is a normal scenario. I mean, it happens in everyone, but yes, I'm not completely certain that that my project covered all the bases in terms of the different situations and different concentrations and ratios in different diseases and conditions. But yeah.
I understand completely. And, unfortunately, summers are short, but, that this work is very exciting, and I'm excited to see where it leads as those comparisons are made. With that, I, again, just want to thank you for your time, and I'm going to turn it over back to Maria.
Thank you.
Wow, that was incredible, Danielle. Her science and her perspective about aging, and Lily, the work you're doing with the education department. We're so proud. Okay, so our next speaker is Dr. Annalise Barron. She's a William Myron Keck Associate Professor of Bioengineering at Stanford University. Her presentation today is titled Anti-Amyloid and Anti-Infective Peptoid Mimics of LL37. You can find her presentation summary and bio on the event's website. And now let's welcome her on stage.
Hello, does everybody hear me? Do you hear me?
We can hear you.
Okay, great. All right, so I checked in with Michael Ray, and he mentioned that so far nobody has talked about the connection between brain infection and dementia or Alzheimer's disease. And so I'm gonna give a pretty good background and introduction to that topic, and then I'm gonna share my lab's approaches. So really we're taking 3 different approaches that probably honestly will all need to be employed to prevent people from losing function as they age, neurologically and intellectually. Once it's fully understood that, as I'm asserting here, Alzheimer's disease, which is becoming more prevalent and seems to have been made possibly worse by the COVID pandemic, is caused by the chronic dysregulation and weakening of innate immunity and the blood-brain barrier via underexpression, degradation, and inactivation of innate immune proteins, including LL37 and apolipoprotein E, which is well known to be a major risk factor for Alzheimer's disease. So the broader title of this would be the role of innate immune dysregulation in the etiology of dementia and how we may be able to prevent and treat it. Alternatively, the SENS-backed approach to doing this is to create anti-amyloid and simultaneously anti-infective peptoid mimics of LL37. So I'm going to summarize the whole lecture right here, just to put everything in context before I continue with the details. So this is work that's funded by an NIH Pioneer Award that I won in October 2020, and it's been supplemented now by funding from SENS. And the basis here is that the human brain is susceptible to invasion by neurotropic, meaning, able to merge with neurons, become active in neurons, neurotropic viral, bacterial, and fungal pathogens. For example, herpes simplex virus 1, for example, the oral pathogen Porphyromonas gingivalis, and for example, the ubiquitous fungal pathogen Candida albicans. Those are all found in the brains of people that died with dementia. So why is it that the brain is susceptible to invasion by these different kinds of pathogens? These organisms, specifically P. gingivalis, also C. albicans, they release enzymatic or peptide virulence factors that degrade and inactivate your key innate immune proteins, including host defense peptides and cytokines. They basically down-regulate your body's immune system. So when you inactivate your body's host defense proteins, and that includes cytokines like TNF-alpha, also the interferon response is downregulated by oral infection by P. gingivalis, apolipoprotein E and LL37, which is a host defense peptide. It'll dysregulate the innate immune system of the whole brain, leave it vulnerable to infection. Once you have infection, you have neuroinflammation. And once you have neuroinflammation, you get neurodegeneration, and you get a steadily declining cognition. Now, I am particularly interested in a human host defense peptide, which is natural in the human proteome and unique in the human proteome, called LO37. And it is broadly antimicrobial. It is also requisite for autophagy and microautophagy. It also, this is extraordinary, it binds to and neutralizes the A beta peptide that's associated with Alzheimer's, the islet amyloid polypeptide that's associated with type 2 diabetes and the formation of amyloid in the pancreas with insulin insensitivity, which is again correlated with Alzheimer's, and alpha-synuclein, which is the particular protein that forms amyloid in Parkinson's. So LO37 actually binds to all of these different peptides in a sequence-specific fashion and makes a complex with them. And we're interested in these complexes. And LO37 is also necessary for the maintenance of tight junctions, both in the blood-brain barrier and the gut epithelial. So it just does so much. And if you have a brain tissue that's left unprotected because LO37 and these other host defense proteins have been degraded, we now have tissue that's vulnerable to phagocytosis, which leads to atrophy. So, literally loss of brain tissue. And we want to prevent this in one of three ways. And honestly, you could probably do all three of these. If you had someone susceptible to this, for instance, by having a apolipoprotein E epsilon 4 allele, you could do all three of these strategies to try to prevent this. One thing I think will be important is that you do want to upregulate LO37 from birth to try to prevent. And I think the best way to do that is with the BCG vaccine, and that's the substance of my NIH Pioneer Award. You can also orally dose a person with compounds that upregulate LO37, and I'll talk about that. And then lastly, when you already have an established brain infection, you can utilize biostable mimics of LO37, compounds we call peptoids, that share LO37's anti-amyloid and anti-infective properties. So I just want to show you, this is the microfiche photograph from Dr. Alois Alzheimer that was published in 1907. And you see how much brain tissue is lost actually in the Alzheimer's disease. I mean, there's just a massive atrophy of the brain. And we're looking at more than 55 million people suffering from Alzheimer's globally and 5.5 million in the US, with only 1 in 4 in the US diagnosed. Until 1984, actually, it was believed that Alzheimer's disease might have an infectious origin. But then that year, they discovered the amyloid beta peptide, and then 2 years later, phosphorylated tau, both of which form amyloid fibrils external to neurons in the case of A beta, and internal to neurons also in the case of A beta, and specifically phosphorylated tau. And, so therefore around 84, 85, 86, this amyloid cascade hypothesis was born, which put forward the idea that dementia and Alzheimer's was really just caused by these amyloid forming proteins, A beta and tau. And I would say that's not wrong, because we know that a rapid overproduction of A beta can contribute to the development of Alzheimer's, but it's incomplete. So more recently, and this is from June 2020, it's been recognized even, by very well-known authors like Anthony Komaroff from Brigham and Women's Harvard, and published in the Journal of the American Medical Association, that, hey, maybe infections can cause Alzheimer's disease. We used to think it was just amyloid beta, tau, and ApoE, as I was just discussing, but now there's a new disease model saying that possibly infection triggers neuroinflammation, and then the sequela of neuroinflammation is the so-called Alzheimer's disease. And then they went through a consideration of pathogens that they believe could be involved. One is herpes simplex viruses 1 and 2. And that's very significant, since at least 85% of US adults are carriers of at least HSV1. So that would mean that you're susceptible. And in fact, adults that have a reactivation of a herpes simplex infection see a substantially increased risk of Alzheimer's disease. In particular, if a person is positive for an IgM, it's a particular kind of immunoglobulin that signals reactivated infection. So chronic infection with herpes, it will almost double your risk for Alzheimer's. Whereas the IgG, which is showing less chronicity, did not have an effect on risk. So chronic infection was a problem. And then here, amyloid beta peptides themselves were shown to be active against the herpes simplex virus in a neuronal cell culture model. This is work done at Sherbrooke University. And they showed that if you combined A beta 42 and HSV1 in culture, you would induce the production of the immune response facilitated by TNF alpha and IL-1 beta and interferon alpha in neuroblastoma and glioblastoma cell lines. And then also they were intrigued to see that the neuroglioma cells, so these are, immune cells, would produce A beta 42 in response to HSV1 infection, and that then inhibited secondary viral replication. And then at Harvard, Robert Moir showed that if you used a 5x FAD mouse model of Alzheimer's, and then you infected that mouse model of Alzheimer's disease, which overproduces A beta, human A beta, into its brain, that a herpes virus infection would greatly increase and accelerate the deposition of that A beta in the mouse's brain. And that further, if you went and you looked inside of those amyloid plaques, you would see that the amyloid is actually entrapping and inactivating the herpes viral particles, which again, would seem to indicate that amyloid is an immune response to infection and has a direct anti-infective aspect. A group out of New York City, the Icahn School of Medicine, did deep next generation sequencing of more than 1,000 human brains. And they compared people that died from Alzheimer's disease with normal brains. And, they were looking for differences in gene expression between these groups. But what they found instead, since DNA sequencing also reveals the genomes of pathogens, that they found a lot more of the human herpes virus strains 6A and 7 in the brains with early stage dementia than normal brains. But they really couldn't tell whether the virus was the trigger of the disease process associated with early stage dementia, or whether it was a symptom of dementia. Has the brain become unprotected? But this was a very strong, again, human brain derived data. In a large study in 2018 in Taiwan, they found if adults were diagnosed with new herpes simplex virus infections, they would have a 2.5-fold higher risk of developing dementia. But if they would accept treatment, antiherpetic medications such as acyclovir or valacyclovir, which is a protease inhibitor against herpes simplex virus specifically, you could reduce your risk down to just 9%. And this was a large study with 33,448 subjects. So there seems to be a large effect. Another group at Tufts University made an organoid model of herpes-induced Alzheimer's disease, and they found that even in normal human brain-like tissue, if they infected with herpes, they got every symptom of Alzheimer's disease: gliosis, neuroinflammation, decreased functionality according to electrical activity. And this was in the absence of any exogenous mediator of AD. All they did is take totally normal human brain cells that they had differentiated from stem cells, and then they infected with herpes. So that is a big deal. And in fact, another pathogen of interest is Porphyromonas gingivalis, and you can infect totally wild-type mice, not mouse model of Alzheimer's, and you will see, again, all of the signatures of Alzheimer's-like disease in mice that you orally infect with Porphyromonas gingivalis. You'll see higher inflammation. You'll see neurodegeneration in the infected, but not the control group. And you actually detect the Porphyromonas gingivalis and the virulence factor called gingipains in the hippocampi of these treated mice by multiple different methods. And then finally, you would see the signature extracellular A beta amyloid and then intracellular phospho-tau tangles in the infected mouse group, but not in the control group. And then there was a 2019 study, again, concerning these Porphyromonas gingivalis and the gingipain proteases, which are enzymatic virulence factors that they make. They looked in human Alzheimer's brains. And again, they saw that these Porphyromonas gingivalis organisms and gingipain proteases were in the brains, that the levels they had correlated with tau and ubiquitin pathology, and that they also orally infected PG mice, but they used a different strain of mice, and they used older mice, whereas the previous study used younger mice. Again, they saw a response of a higher production of A beta upon infection. They also showed that the gingipain proteases were neurotoxic, both in vitro and in vivo. So P. gingivalis is interesting because it's an anaerobic bacterium, but it only survives by eating peptides, proteins, amino acids. And so it secretes 3 different variants of these powerful cysteine proteases called gingipains that cleave either at arginine or lysine. And then it also makes a virulence factor called peptidyl arginine deaminase, which again alters arginines. That same group at Cortexime, led by Stephen Dominy, who's now working in my lab for the last year, also showed in cell culture with infected neurons that, you will have neurodegeneration like in Alzheimer's, and the persistent expression of active gingipains that then dysregulate the neurons. So, that previous paper I was just talking about, they actually found these gingipains in 90% of postmortem Alzheimer's brains, gingipains localizing into the cytoplasm of neurons. And then when they did the cell culture study as a companion research project, they showed that PG caused a time-dependent infection-induced cell death, and that further the PG would invade and persist in the mature neurons. And if they then examined the ultrastructure of these neurons, they would see autophagic vesicles, multivesicular bodies, cytoskeletal disruption, more phosphorylated tau as opposed to normal tau, and loss of synapses. And I just want to go back and, like referencing the student talk at 11, right? Senescence and cell death or failure of cells to divide are very likely connected with this chronic infection, especially because cell division is powered by mitochondria. And if you look at mitochondria within the neurons of people who died with Alzheimer's disease, you see a whole lot of mitochondrial damage. And when you use electron microscopy with nano gold that's been labeled with antibodies against these arginine gingipain proteins that are derived from P. gingivalis, you actually see that inside these damaged and dysfunctional mitochondria, in fact, the gingipains are present. So a major reason you would think, a cell that lacks the mitochondrial energy production capability, that it needs will not be able to divide and will go into senescence. So I think that is a major cause. And they actually have known for quite some time that mitochondrial dysfunction can be caused by these outer membrane vesicle OMVs that get released by pathogens. So here's just gram-negative bacteria, Neisseria gonorrhea, uropathogenic E. coli, right? Some of you may be aware that bladder infections are caused by uropathogenic E. coli, and bladder infections cause delirium and dementia-like symptoms. And Pseudomonas aeruginosa is a common bacterium. These all can damage mitochondria, and that would lead to senescence. And if you look at genome-wide association studies, you'll see that multiple host pathogen interactome pathways are upregulated in Alzheimer's brains. And here's really the serious issue that we face as a human race. We're not going to be able to vaccinate against every single kind of pathogen that can appear in these brains of people with Alzheimer's disease. So I've listed, this is a short list of things that have been found in these Alzheimer's brains: Candida albicans, Cryptococcus neoformans, Bornavirus, Borrelia burgdorferi, which causes, I'm trying to remember, it'll come to me in a second. Cytomegalovirus, roseola viruses, that's the HHV 6A and 7, herpes viruses, HIV, Epstein-Barr, which is also correlated with multiple sclerosis, hepatitis C, influenza, P. gingivalis, which I just spent some time talking about. Also, I just am going to mention that there's a spirochete bacterium that causes syphilis. And it's been well known that syphilis caused dementia and delirium, since forever. HIV causes that same neurocognitive dysfunction, as can influenza, hepatitis C, and COVID, right? Now, the strongest risk factor for Alzheimer's dementia is apolipoprotein E, epsilon 4 allele. And in fact, ApoE plays a role in activating immunity to infection because ApoE needs to bind to the activated innate immune protein C1q. And that complex then kicks off complement, and complement is how you kill infected cells. And I just want to show you, this is a skull of a monk that had syphilis, chronic syphilis. And these infections can even degrade bone. That's these holes you see. So, degrading brain tissue is really not a problem. So, and in fact, if you just paint only these gingipanes onto the gums of young 8-week-old wild-type mice, and you do that for 22 weeks, 3 times per week, you actually kill half of the hippocampus in about half a year. And the folks at Cortexheim, so Steve Dominy gave me this data because he works in my lab now, they showed that these gingipanes actually preferentially chew up and inactivate ApoE epsilon 4 as opposed to epsilon 3 and epsilon 2. So these are the only differences between the 3 common human alleles of apolipoprotein E, 2, 3, and 4. 2 is extremely protective, 3 is somewhat protective, and 4 is a very high risk allele. And these gingipanes actually cleave at arginine. And what the Cortexheim group showed is that ApoE epsilon 4 becomes inactivated by alterations and cleavage of those arginines. Further, the peptidyl arginine deaminase enzyme that's made by P. gingivalis will convert arginines to citrulline, and that will inactivate this protein I'll talk about in a moment, LO37. And it does that by citrulinating arginine. And it's well known that the hippocampi of AD have an abnormal accumulation of citrulinated proteins. So let's see, I have 7 minutes left. So a logical deduction would be if dementia and even neuronal senescence is actually caused by infection, and by the way, your odds of having P. gingivalis infection by the time you're 55 years old is almost 100%. And your odds of having herpes simplex virus 1 infection as an American adult is 85%. And so by the age of 60, you're very likely to be colonized both by P. gingivalis and HSV1. And then it's just trench warfare. How long is your brain going to last against this onslaught? If you're going to have multiple different pathogens, there must be some ongoing dysregulation or dysfunction of the brain's immune system that would seem to be responsible for this. How does that happen? So, I really believe that we have a destruction of the proteins that would normally defend your brain, so-called innate immune modulatory factors, apolipoprotein E epsilon 4 and epsilon 3. Those 2 can get degraded to a significant extent by the gingipanes. and other virulence factors. And also this important immunomodulatory host defense peptide LL37, which is necessary for controlling innate immunity. And LL37 is a kind of lever to shift innate immunity. So one thing you might think about doing is upregulating the gene that encodes LL37, which is a 37 amino acid peptide that is one of a kind in the proteome and also in the rodent genome, encoded by a gene also called CAMP. So it's only one gene, one protein. And it's considered to be the most centrally important or factotum host defense peptide in the human system, because it has the attribute of being extremely broad spectrum antibacterial, antiviral, and antifungal, and prevent the formation of stable colonies of pathogens called biofilms. And it can be deployed from all different kinds of white blood cells, neutrophils, macrophages, microglia, which are the white blood cells of the brain, dendritic cells, which are the immunomodulatory cells of the body, and then specialized immune cells like basophils and eosinophils, T cells, and natural killers all utilize it. But also neurons can express it, platelets, mast cells, the meninges, which forms a protective barrier around the brain, and all types of epithelial and endothelial cells, which are, again, important. And this peptide is released by a dedicated proteinase. It's necessary for macroautophagy and maintenance of tight junctions. The problem is it has many arginine and lysine protease sites, and we already have established that these gingipanes are, very dangerous to peptides and proteins that have arginines. And there's also a lysine protease among the gingipanes. So they get rapidly degraded, and so we lose all of these important pleiotropic properties of LL37, its antimicrobial activity, its ability to recruit mesenchymal stem cells, which would allow renewal of brain tissue and other tissue, nerve tissue. It's supposed to induce apoptosis in cancer cells, immunomodulation, wound healing, and angiogenesis. All these are really important, including autophagy, and we lose those functions. So LL37 and A beta, which are both regulated by the same two G protein coupled receptors, the formula-like peptide receptor 1 and the P2X7 receptor, our lab showed in 2017 that these form a complex with each other. And in fact, the common familial mutations in Alzheimer's disease would tend to disfavor the formation of those complexes, because most of those amino acid sequence alterations that predispose to Alzheimer's actually reduce A beta's negative charge, and therefore would be expected to reduce the electrostatic interactions with the positively charged LL37 protein. My lab showed that LL37 is expressed in all organs of the body, with the highest levels in the brain and the GI tract, that it's upregulated in the case of infection, that LL37 can be neuroinflammatory, but LL37 strongly inhibits A beta fibril assembly and shows sequence-specific binding with A beta, which is the amyloid associated with Alzheimer's. It binds to oligomers more strongly than fibrils, so it would tend to prevent the formation of fibrils. And it prevents LL37 binding to A beta will prevent the formation of beta type secondary structure. And that binding then protects against the toxicity of A beta and the neuroinflammation it causes. So working with Yevgeny Raskatov, originally we examined the kinetics of LL37 A beta binding using the fluorescent molecule THT, which binds to amyloid. And we showed that an equimolar mix of A beta and LL37 completely abrogated the formation of amyloid, whereas if you had less LL37 than A beta, you would just slow the formation of amyloid, right? So you could totally abolish it if it was 1-to-1 and slow it if you had less than 1-to-1. We later went in and showed this complex is unique using amyloid analytical ultracentrifugation. Josefina Nielsen in my lab used small angle X-ray scattering and showed that this interaction is unique, that these perform defined nanostructures. Circular dichroism showed no amyloid formation. And we showed that, in fact, it really, even if you look just at A beta 40, a shorter version, you really prevent the formation of amyloid with LL37. and I'm running out of time here. But I will just say that what we are doing in the lab now is recognizing that LO37, even though it has this advantage of complexing with A beta on this millisecond time scale, and we saw this by stop flow, it has the disadvantage that it'll be degraded. And so what we have done is looked at biostable mimics of LO37, in particular this peptoid 1, and we showed that these compounds, which are also extremely broad spectrum antimicrobials, and they are antibacterial and antiviral. And if you look on my webpage, you'll see so many publications about these antimicrobial peptoids. If you look at this orange line, what we see is that these peptoids also completely block the formation of A beta fibrils. And so what we're doing, and we're in the midst of doing this right now, is further, we're looking for novel peptoids. And I have a student named Christian Sorensen who's making new peptoids, which we also have found bind to LO37, although so far that peptoid 1 is still the best one. And actually, I am going to just stop here because it's 11:46, and I'll just say if this underlying hypothesis is true, the human body already has an endogenous drug that can prevent amyloid formation and infection, but it will fail to do its job in the presence of these, these pathogens that can cause degradation of LO37. And so our best idea, if we can't prevent PG, is we can mimic LO37 to try to clear the infection. So I will actually stop sharing now. And I'll say thank you for your attention. And I hope this was interesting and surprising to this group of speakers.
Very, very interesting, Annalise. I'm sure everyone agrees. Yes, they're sending their hearts and likes. And
thank you. That's fun to see all the hearts.
Yes, so remarkable. Thank you so much for being with us, Annalise. Are you gonna stay around?
Yeah, I'm staying.
Okay, so I wanted to tell everyone, because we have a few questions in the chat, but we don't have time to answer them here on stage. So they can find Annalise. You can find her by searching her name in the chat, and then you can call her from there. So you can kind of abduct her.
I'll take a look. I see the questions.
Okay, wonderful. And then people can find you like that using the chat. So thank you so much again.
Thank you very much.
Thank you. So everyone having a good time? Show me with your fingers and hearts. Yay, okay. Are you enjoying the platform also, the metaverse? Okay, cool. All right, so we're gonna go with our next presenter, and it's Dr. Ivan Schneider. He's a professor and director of the Center of Stem Cells and Regenerative Medicine at Sanford Burnham Medical Research Institute. His presentation is called, "Is there a common neural network signature to cognitive disorders that is druggable? A stem cell-based approach." And you can find a summary of his presentation and bio on our website, on the events website. So please, Ivan, come to the stage.
I'm not as familiar.
Welcome. I'm still on stage. Ooh, I shouldn't be, but I'm still here.
And I don't have control of my presentation. I don't.
Okay, let's see how we fix that.
How does that look?
Is it getting better? We can see it.
No, you shouldn't be seeing that.
Something's definitely happening because I'm still on stage, but so we'll see.
What are you seeing now? Are you seeing presenter view or are you seeing my slides?
I see your slides. I don't see presenter view. We see everything. Let's see.
What do you see now? Do you see Mitch McConnell?
Now, yeah.
But
now it looks good with presenter mode.
Do you also see my notes?
No, I don't see your notes. It's good. You're good.
You're just seeing the slide, right?
Yeah. Wait, that's good.
An answer to your question is do I like this platform? It depends on how my talk goes.
Let's see.
I can't see me at all, but as long as you can just see the slide, not my notes, and just the title, then we're good, right?
Yeah, we see you and they see me. I'm going to try to get off stage here doing something different because I shouldn't be on stage. But everything's good. You're good to go. You're good
But
to
just
go.
clarify, you're just seeing my slide. You're not
Yeah.
seeing the next slide. You're not seeing the notes or anything like that,
We
right?
see a slide. It says, "Is there a common?" It's just the title of
Okay,
you. Yeah.
great.
Everyone can confirm that with me by yeah. Okay. I see different stuff from here than what the audience sees. Okay, go for it.
All right, thank you. All right, well, it's really an honor to be asked to speak at this conference, particularly because, as most people know who have heard me speak, I am not a gerontologist. I'm a pediatrician, actually. But and I guarantee I'm not going to go over time. I'm going to talk fast because I'm about to be kicked out of my hotel room. I'm at a graduate student retreat, and I'm already overstaying my welcome here. Anyway, what I'm going to be talking about is not so much curing or blunting aging, but talking about some of the cognitive problems and trying to understand them and reverse them once we know that we have them or they're developing. I want to play this video that I think many of you have seen in the news, and I'm going to comment on it throughout the talk. Hopefully you can hear this. So,
running for re-election in 2026.
Did you hear the question, Senator? Running for re-election in 2026? All right, I'm sorry, you all. We're going to need a minute. Senator, please.
Okay.
Somebody else have a question? Please speak up.
okay, what Mitch McConnell was having there is an absence seizure, but it's not the kind of seizure that we're used to seeing. There was not motor activity. There was a cognitive pause. And what I'm going to make the argument and what I'm going to describe, and you'll see this at the end of the half hour, is the cognitive pause that he experienced through hyperexcitable neurons. Now, his was a seizure, but non-seizure conditions will cause that cognitive lapse. And how you get to the hyperexcitable neurons, that can vary. But once we have them, I'm going to make the argument that these could be druggable. Now, this journey that I'm going to be talking about did not come starting looking at aging. And as you'll see, it emerged as the work began to unfold. Now, you heard that I'm a stem cell biologist, and one of the jobs of stem cell biologists is to model diseases. And the way we've come to do this over the past 15 years is to take somatic cells from a patient with a given disease and then to turn them back into stem cells, emulating what you might see in the blastocysts. And depending on the organ of interest or the cell type of interest, one replicates developmental processes to give you these various cells. So I'm going to be talking about nerve cells. Now, what's interesting is that, and this is a bit of a conceit of the stem cell biologist, is that these neurons that have been derived from blood or from skin from a patient actually have that disease. Now, it's a bit of a stretch, and as I said, it's a bit of hubris to say this, but it looks as if this has turned out to be the case over the past 15 years. Now, most of the time, IPS lines are used to model monogenic diseases. However, we became interested in determining whether they could be used to understand complex polygenic multifactorial diseases, not just describing the phenotype, but actually getting to the molecular mechanism. Neuropsychiatric disorders are the poster child for those diseases. But we decided that we were going to take neuropsychiatric disorders And imagine that these are inscrutable. And that's kind of this can, and we need to get into the can. And we decided that we would pick probably the most heritable psychiatric disorder that exists, bipolar disease, which is also incredibly prevalent and highly lethal. But what's unique about bipolar disease, and seemed to be our opening wedge, our molecular can opener, so to speak, is that a third of the patients respond to lithium for unclear reasons, but it absolutely reverses the phenotype. But again, it's not known. We reasoned that if we could figure out lithium's target, its protein target, we could take that anchor and then map upstream and downstream and find what is the cause of bipolar disease. Again, a little bit of a stretch, but it seemed to play out. It turned out that lithium's target is a molecule called CRIM2, which is a master cytoskeletal regulator. I'm not going to go through all of the data that was published in this paper of a few years ago, except to say that when you look at all the protein that are altered in response to lithium and you do a map, they focus on axonal guidance signaling. And the regulator of that is collapsing response mediator 2, or CRIM2. Now, why is cytoskeletal regulation important? It sounds as if it would be very distant from cognitive problems. But if you think about it, ultimately everything focuses on whether connections are made from axons onto dendritic spines. And of course, cytoskeleton regulates ion channels and things of that sort. And again, without going through all the data, what we found is that CRIM2 is in an activated or inactivated state. When it's activated, it binds cytoskeletal elements and allows dendrite outgrowth. When it's inactivated by being phosphorylated, it releases all those cytoskeletal elements and retracts. Now, this is a normal process in all of us, and we all toggle back and forth between these states in response to what's going on in the brain: inflammation, lack of inflammation, changes, growth, things of that sort. In the bipolar patients, the lithium responsive bipolar patients, the set point is just too high. The ratio of inactivated phosphorylated CRIM2 to unphosphorylated or total CRIM2 is just off. And what lithium does is re-equilibrate it. Now, why is this important? The business end of neurons basically is how they make network connections, how they make synapses. The synapses are on dendritic spines. And what CRIM2 does, as you can see over here, is that when it's active, it goes into the spines. When it's inactive, it retracts from the spines. And it's a dynamic process, on and off, and responsive to external cues. So if you focus on how important dendrites are, and you figure CRIM2, among its many roles, is pivotal in how these dendritic networks look, you, of course, then try to test that. So we went to a mouse, we knocked it out. And of course, what happens, you can see, is dendritic spines kind of become diminished in number. And in fact, the entire dendritic tree is aberrant. That's in a mouse. If you go to a brain bank and look at the brains of bipolar patients, you can see the same thing, a true dearth of dendritic spines. Now, what's interesting is the ratio is abnormal in these patients. Now, keep track of this because I'm going to come back to this later on in the talk about this abnormality in the ratio. This is in one brain bank. We went to another brain bank, and the same story is true. The ratio is abnormal, too high in activated CRIM2, and the dendritic trees are aberrant. The spine density is diminished unless the patients have taken their lithium, in which case things normalize. If you look at the activity of the neurons, and one can do that by looking at calcium kinetics, one can see that the neurons, and I'll come back to this theme again, are hyperactive. Now, they are hyperactive only in the lithium responsive bipolar patients. and very rapid ingress and then egress of calcium. But lithium normalizes it. Also notice that lithium non-responsive bipolar patients have a normal curve. This is another topic, but probably lithium non-responsive bipolar patients are an entirely different disease. So I'm going to focus on pure true bipolar disorder, which is lithium responsive. Now, lithium does not help in a mouse in which CRIM2 has been knocked out, as I showed you before, because lithium's target has been eliminated. Now, to show that we really were looking at exactly the residues, exactly the point where lithium was working, there was a mouse in which we prevented CRIM2 from being phosphorylated. And one simply does that by changing one of the residues. So now you have a CRIM2 that basically cannot be phosphorylated and is excessively active. So this is hyperactive CRIM2, which is not abnormal in and of itself, but it allows us to genetically mimic what lithium should be doing. If you then put that into one of the behavioral assays for lithium-responsive bipolar disease, which is you take a wild-type mouse, you give it methamphetamine, and a normal mouse with a normal CRIM2 just goes nuts. And you can see spends very, very little time in the unprotected center exploring it. It goes manic. Lithium will change that. So now we take the two mice, a wild-type mouse and this non-phosphorylatable CRIM2 mouse, a mouse who should act as if he's on chronic lithium. You put them in this open field, you give them methamphetamine, the wild-type animal goes nuts. The non-phosphorylatable CRIM2 animal acts as if he's on chronic lithium. Nothing changes. So this reinforces this notion that we really have nailed down exactly what lithium is doing in CRIM2, and CRIM2 is important for this. Now, this is just a histogram showing the data for that. Now, this ratio can be used for drug screening. What changes this and normalizes this ratio? Now, we can go downstream of lithium. We can find, now that we know the lithium response pathway, we can be more incisive and more surgical. Lithium is a bad drug. It just has too many off-target effects. But now we can look at what lowers CRIM2 and things that could be hitting all these various aspects of drug action. You do it through high throughput screening. And what we found, in fact, that there were a few other FDA-approved drugs that could mimic lithium, that in that it would diminish or eradicate this manic anxiety behavior that I mentioned before. I'll come back to this, but it's interesting. Huperzine is one of those drugs. People that might be familiar, huperzine is a drug that's been an ancient Chinese medicine. It's a natural product that has been used for centuries for dementia and cognitive abnormalities. Another thing that is interesting is that patients with lithium-responsive bipolar disease have a higher incidence of dementia and Alzheimer's disease, unless they've been compliant with their lithium throughout their lives. Now, we think of bipolar disorder as a mood disorder. I want you to listen to this video of a patient with bipolar disorder. It's going to be a little different than Mitch McConnell, but you're going to see that there's a cognitive disorder here.
Why you're here and why I'm here. I'm the psychiatrist in the emergency clinic today. Right, and your GP's asked me to see you.
So that's what I mean.
Is that right?
Yeah, because I've got to tell you about this.
Okay.
Yeah,
okay.
a cure for cancer, isn't it?
Cure for cancer.
Cure for cancer.
Okay.
Yeah.
How's that going to work then?
Well, how it works is God tells me, that I'm doing a really good job,
Right.
how great I am, and how fantastic it is I'm working at this,
Okay.
and I just keep going at it. I
Okay.
don't stop.
Okay.
Yeah.
Can I just take you back a step?
Yeah,
You
sure.
said God tells you.
God tells me,
Can you
go on.
tell me a bit more? It's all been keeping you really busy.
Really busy, really busy.
Have you been sleeping at all?
No time to sleep.
No time to sleep.
No.
When was the last time you had a good night's sleep, would you say?
Don't need it.
Don't
I
need
need
to.
it. I've got this
Okay.
enthusiasm from God, you know. It's
Right.
there, so there's
So what's your
energy
en-
inside me. That's
So you feel as though you've got a lot of energy?
Yeah.
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 And how are your
Tons of it,
thoughts?
tons
Are
of
they
it,
are
tons of
they
it.
are they going fast? Are you thinking
Well,
lots?
yeah, because I have
lots
to,
of
I have to
ideas?
think very intensely. You know, I mean,
Right.
you look at stuff down here. I mean,
Yeah.
there's not. Yeah, I got that in the door the other day. You know,
Okay.
and there's lots of numbers of these.
I guess, John, what I'm wondering is whether this might be a sign that you're not very well at the moment.
No, I'm fine. I want to talk about this. You seem to want to change the subject, so I
Yeah.
don't want to talk to you anymore. I've had enough of you, and I've had enough of being out. I want to talk to somebody else. I want to talk to the boss. I want to talk to your boss. I want to talk to the top man, top boss, top cat. That's what I want to talk to. Top cat, top cat, top cat. Yeah.
Now, this is a patient with bipolar disorder, but I think you can see it's not just a mood disorder. There's a true thought disorder. There's a poor connections between various things. You don't see it the way Mitch McConnell had it, but there are abnormalities in thought and processing and cognition. Lithium normalizes that in that kind of patient. And just about the time when we published this paper, some other interesting data started coming out. Tens of thousands of psychiatric patients were analyzed simply based on their genomic expression. And this is about 6 separate studies. And it was found that lithium-responsive bipolar disorder does not cluster with mood disorders. It actually clusters with cognitive disorders like schizophrenia. But now this seems a bit counterintuitive. I told you that the neurons are hyperexcitable, and we talk about patients being manic. We don't talk so much about their thought disorder. We talk about a mood disorder, but that's not what the genes are telling us. So we decided that we would look at that. And in fact, as you might expect, and as I suggested before, the more inactive CRIMP2 is, and the more phosphorylated it is, the more hyperexcitable, the more hyperactive are the neurons. Those were in the mice. This is now human bipolar neurons derived from IPS lines. Lithium normalizes it, and it brings it to a level that is normal in a normal patient. So now let's look at the networks, and you can do this through multi-electrode array, where you look at how each individual neuron that's spiked connects with each other. And rather than there being hyper-functioning networks, as you might expect from hyperexcitable neurons, actually what you can see over here is that the networks are actually fairly impoverished. Things are working worse in terms of their networks. Now, why might that be? Well, a prosaic explanation might be simply that the spines are diminished. But we're not interested in actually what does the neuron look like itself. How do they link together? So what Cameron Pernia, a grad student in the lab, is he looked at the neurite outgrowth, pulled them out, and did proteomics on those, particularly looking to see what does CRIMP2 regulate. And he put together this kind of network, this network diagram. And of course it regulates cytoskeleton, but the highest category of pathways that it regulates is actually calcium activity, calcium signaling. These are the neurons that you see from a bipolar patient. Notice everything is firing at the same time. This is something called synchrony. It's never been determined to exist in psychiatric disorders, in neurodegenerative disorders. It's known to the epilepsy field, and I think you can see where I'm going. Everything is on, everything is off. This is what a normal picture should look like. Hopefully you're able to see this, but you can sort of see a little bit of activity in one area, a little bit of activity in another area, a little activity, not everything on. That's how that's how signaling takes place. And if you couldn't see it from the actual calcium imaging, here's what a graph of it looks like. This is what normal activity should look like. This is synchrony. And what's interesting is the greater the hyperexcitability of the neurons, the poorer they hook up together because they create synchrony. This is what we see with mouse neurons from the knockouts and the knock-ins. This is what we see in actually bipolar neurons, which you can see over here, increased syncope. So counterintuitively, the more excitable the neurons, they actually make for less effective neural connectivity, circuitry, and networks. And the way I like to tell people to think about it is the way you get information, let's say from a screen, is you need dark pixels on white pixels. That's what gives you forms, or white pixels on dark pixels. If everything is white or everything is black, you get no transfer of patterned information. And that seems to what's be happening there. Now, if you normalize CRIMP2 activity, and you do it either genetically or you do it with lithium, now information transfer improves. Does this have any relevance to the human condition? Well, if you look at fMRIs of bipolar patients, here's what a normal patient looks like. Again, little activity here, little activity over there. In a bipolar patient, everything is firing at the same time. There's poor information transfer. Now, we know that hyperexcitable neurons characterize many neurodegenerative conditions, including Alzheimer's disease and ALS and Parkinson's. Calcium dynamics seem to be impaired. With that in mind, we decided, well, we know with Alzheimer's disease that there is a deposition of a cytoskeleton. Tau is a prominent one. If CRIMP2 is abnormal, what could tau be doing? And sure enough, if you go to the brains of an Alzheimer's patient, in the plaques, there's an accumulation of phospho-CRIMP2. And they're kicking me out of the room, but let me continue going quicker through here. Also, in fact, you can see that the ratio in Alzheimer's patients is increased, the ratio that I told you before. But in the bipolar neurons, you can see that APP is upregulated. That's abnormal. If you increase CRIMP2, you can see that the various aspects that we know about that are relevant to Alzheimer's disease are normalized. And lithium decreases the amount of dimerization of tau, and also the decreases the phosphorylation of tau. If you look at gene expression, there's an overlap between Alzheimer's and lithium-responsive bipolar disease. The overlap, interestingly, is in ApoE, which we know is a high risk factor for Alzheimer's disease. And ApoE in kids has been found to be associated with cognition. We've determined that many of the neurons that we get from patients with cognitive disorders, not just bipolar disease, but cognitive disorders, are hyperactive, including from Alzheimer's disease. Now, why would this be a problem? Imagine, and let's go back to Mitch McConnell. This is a region of hyperexcitability. Now, it could be anywhere in the brain. With Mitch McConnell, it was in a particular area, but imagine it at different areas. If you get poor transfer of information superimposed here, it's not just that particular area, but it's everything going through that area. And it could be trauma, it could be plaques, it could be a seizure focus, it could be degeneration. Imagine what happens to make it more palpable to what we've all experienced. Chicago is shutting down in a snowstorm. It's not just Chicago that shuts down. It's transfer of information, transfer of flights all throughout the brain. Now that we have with calcium dynamics able to be quantified, we can now begin to use machine learning to begin to understand, make diagnosis, and even use this for drug discovery. In bipolar disease, the separation between calcium handling and the neurons is so discrete and so separated that we could teach a machine to diagnose lithium-responsive bipolar disorder, as well as determine whether the patient would be responsive to lithium. We could begin to do the same thing with cognitive disorders, simply looking at calcium dynamics, determining Alzheimer's disease from normal neurons, Parkinson's disease that have cognitive defects from normal. But you could say, well, even my grandmother can tell whether a patient's normal or not and distinguish that. The calcium dynamics can distinguish various kinds of cognitive disorders, which hopefully will give rise to a clinical tool that we can use not only for diagnoses, but maybe for drug discovery, not just biochemical assays, but actually functional assays. The key is going to be distinguishing between the various kinds of cognitive disorders. So I'm going to end here by saying we're not going to be curing Alzheimer's. We're not forestalling aging. How one gets there, how one gets to hyperexcitable neurons depends on the understanding of the pathogenesis of those particular diseases. However, we think that if we can understand the functional defects, we may be able to counteract, forestall, prevent, reverse, or compensate for the cognitive decline, which means that what I've begun to embrace is the notion that diseases of the brain, including those of aging, are what I call networkopathies. So the concept of networkopathies, I think, is really what underlies cognitive disorders. And in fact, in regenerative medicine, we got it wrong in talking about new neurogenesis and cell replacement or cell protection. It's the recreation, the preservation of networks, which I think are important. Counterintuitively, hyperactive neurons actually make for hypofunctional neural networks. Synchrony is the result of hyperexcitable neurons hooking up with each other. And I think this ties many of the diseases of cognition together. I think disease in the dish modeling can give us mechanistic insight, even when you do not already going into it know the particular gene, cell, region, or whatever. Reverse drug discovery, which is basically what the molecular can open approach is, I think can be used widely. I think that the stem cell field can give us circuit level, give us insight into circuit level pathology. Cytoskeleton is important. Proteomics is important, sometimes even more important than gene expression. I told you a case where it was not the gene or even the protein that was abnormal. It was the post-translational regulation and modification that seemed to be abnormal. And machine learning, I think, at least when it comes to calcium kinetics, can help us with diseases of cognition, including those that result from aging. And with that, I'll end.
Oh, I'm sorry. I feel bad because I know they are kicking you out of your hotel room. You said, I don't want to kick you out of stage too. Yeah, we're out of time, but thank you so much. That was amazing. Yeah, everyone, show your appreciation. Yeah. So, okay. Well, thank you, Ivan. Thanks so much. And we hope to have you again soon and keep sharing your important research information with everyone. We're closing. Our talks are ending. This is they come to a close. I want to give a big shout out to our hardworking team, founders and directors. Their hard work has helped put rejuvenation biotechnology on the map. Thanks to them, what SENS Research Foundation started in 2009 has grown into a full-fledged field today. I remember when we first started, a lot of people thought our goals were a long shot. And yes, they called us crazy. Now turning back the clock on aging isn't just a dream. It's real science that people take seriously. And we know how hard we worked for this to happen. No matter what stood in our way, we never stopped pushing forward, and we are not planning to stop. We're always looking ahead, and with the driven team that we have, I'm sure SRF will keep leading the charge against aging. A huge thank you to our donors on behalf of the foundation. You're not just supporting us, you're helping shape a better future for everyone. We can't thank you enough. I also want to thank our media partner, Lifespan.io or .io, for being always behind us. And a special thanks to Jessica Blair, Gary Abramson in Linea from Metaverse for the hard work you put into making this event happen. I could have never done it without your help. We hope you had a good time today and yesterday, and we can't wait to see you again. Stick around now, explore and meet others who believe in what we're doing and our mission, the mission of eliminating the horrors of aging and achieving a long, healthy life for everyone. So thank you again. Keep the conversations going. There was a really interesting interaction in the chat about what Dr. Schneider was speaking. I want to see circles formed around the room, and you can keep those conversations until 1. We're here until 1. So thank you again.